Hello, everyone, and welcome to today's webinar, Tissue Preparations and Handling for Multiplex Imaging in Spatial Biology Research. I am Kaylee Bach of Labroots, and I'll be your moderator for today's event. Today's educational web seminar is presented by Labroots and brought to you by Thermo Fisher Scientific. To learn more about our sponsor, please visit thermofisher.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This presentation is educational and thus offers free continuing education credits. Please find the Continuing Education tab at the bottom of your screen and follow the link to obtain your credits. Now with that, I'd like to welcome our speaker, Nancie Mooney, MS, Scientist for Research and Development at Thermo Fisher Scientific. For a complete biography on our speaker, please see the Presenter tab on the right-hand side of your screen. Nancie, welcome. You may now begin your presentation.
Hi, I'm Nancie Mooney. I'm a scientist in research development at Thermo Fisher Scientific. Today I'm going to be presenting about tissue preparations and handling for multiplex imaging in spatial biology research. Spatial biology is an overarching term for the different technologies that allow the overlaying of high-plex or omics data onto tissue imaging. This includes what we consider spatial omics and allows us to take things like epigenomics, transcriptomics, proteomics, and apply them in a more spatial context. This allows us to do things like identify which types of cells are present in the tissues, their biomarker expression patterns, and how they organize and interact to influence the tissue microenvironment in normal and disease state tissues. This is a rapidly growing field with an ever-evolving methodology. So while spatial biology enables researchers like us to characterize combined molecular image into spatial resolution in context, it can complicate our experimental setups and our detection strategies. So, multiplex detection for tissue imaging is a spatial biology technique which allows multiple biomarkers to be simultaneously stained in a single tissue section. So, taking intact tissues and detecting biomarkers like proteins, mRNA, and DNA with detection strategies like chemical stains, antibody detection, and in situ hybridization helps us to elucidate the spatial relationships among individual cell types in these tissue niches. Multiple detection modalities are possible, like I said, in a single tissue section, and sometimes we can do things like cyclical detection and imaging to really allow us to increase the plex of our detection strategies. In this example of multiplex detection in tissue, this is a human tonsil. We have stained it with multiple different antibodies that were conjugated to fluorescent probes. Here we are looking at Ki67, a marker of cell proliferation, as well as pancytokeratin and smooth muscle actin. We have also counterstained using a chemical to identify the cell nuclei. So what this allows us to do is look at the various spatial areas in this tonsil and identify, for example, where proliferating cells are, as well as the more smooth muscle actin rich and cytokeratin rich areas in this tonsil, for example, in the crypts. In this other example of multiplex detection in brain tissue this time, we have actually used two different detection modalities. One is fluorescent in situ hybridization using Thermo Scientific's ViewRNA tissue assay kit. We have used probes against synaptopodin, cannabinoid receptor 1, and polar 2A, detecting those specific mRNA transcripts in this tissue. We have also used a NuN antibody to identify brain cells in this tissue. So this allows us to look for not only the neuronal cells, but see what transcripts they're actually expressing. So a typical workflow for detecting such biomarkers using imaging in tissues is, of course, you need a microscope, and then you have various stages of tissue preparation, antigen retrieval, blocking background, and then, of course, your target detection. You then image capture and data analysis. So Often, if you're doing some kind of cyclical detection, you'll actually end up image capturing and then going back for a second or third, or who knows how many rounds of target detection. Today, my talk is mostly going to be focusing on these earlier stages and a little bit less in terms of target detection specifically, but more of the pre-steps that you need to consider in order to get your best target detection in your tissues. So multiplexing can become very complicated and requires multiple aspects to consider: how the various detection modalities might interact with each other and choosing appropriate treatments in the appropriate orders. Order can absolutely matter. If you're doing something like in situ hybridization, trying to detect individual mRNA transcripts, it's possible that if you're also going to use an antibody, the blocking steps that are best for that antibody detection might result in poorer signal in your in situ hybridization. So order matters. That can take something that's relatively simple, like a single-plex antibody experiment, that's almost a checkers-like situation where you go step by step relatively straightforwardly into chess or even 4D chess, as you kind of need to look forward at your future moves to establish what's your best move at the time. So again, target detection informs what kind of blocking background or what kind of sample preparation you'll do, but also the kind of sample preparation you can do might affect what detection modalities you'd like to work with. So in today's seminar, I'll be going through working through several of these considerations so you can better understand things to improve your experiments. We'll be looking at tissue considerations, fixation and handling, some of the considerations you need for FFPE tissues, epitope and antigen retrievals techniques, detection modalities, and how they might influence the appropriate blocking and quenching techniques, how to deal with some autofluorescence, what mountains you might use at the end of your detection experiments, and some appropriate controls. Again, there are many considerations to make, and I'm hoping that I can make this seem much less confusing. So, to start out with, some tissue sample considerations. In terms of tissue type, some things we should be considering are honestly the thickness of the tissue on your slides. If you're going to be using commercially available tissues, those are often only 5 to 8 microns thick. But if you're preparing your own tissue, they can be as thick as a couple hundred microns. With that being true, sometimes you should be considering how deeply the reagents can actually penetrate and whether there's some strategies you can use to improve that. Other things to consider are the tissue makeup. Is your tissue very sturdy or is it a little bit diaphanous? For example, lung tissue can be very difficult to work with because there isn't a lot of tissue to it, especially around the alveoli structures. Other things to consider is the tissue especially fatty? If there's a high lipid content, you might want to consider a detergent step in order to remove some of the lipid so that your water-based or soluble reagents won't be interfered with by the fatty tissue. Things we'll consider more later, but I would like to mention now as well, is the autofluorescence characteristics of your various tissues. Blood-rich or collagen-rich tissues, or really anything with a lot of repeating structures, can have very strong autofluorescence characteristics. So if you're considering a fluorescent detection modality, you should consider some kind of autofluorescence like quenching. We will also be talking about tissue fixation type in an upcoming slide. In terms of tissue preservation, there are 3 common methods in the scientific literature. One is formalin-fixed paraffin-embedded tissues, known as FFPE, where these are formalin-fixed and then embedded in paraffin for easier sectioning. These often require a little bit of extra handling to remove this wax and to retrieve the epitopes that can be hidden by this fixation method. Another tissue preservation technique is cryopreserved. These can literally be fresh frozen, which is literally snap frozen with no other chemical intervention, or sometimes fixed and then frozen. These are then often embedded in a compound like OCT to allow for better tissue sectioning onto slides. Some experiments can use what we would consider fresh tissue, but often those are also fixed in situ using some kind of perfusion mechanism. These can also need additional fixation to allow the tissue to survive staining, but that can involve some pretty complex steps. So I'm not going to go into this in great detail, but there are a lot of really good resources online that I suggest you look into if you're thinking of using thicker or considered fresh tissues. Okay, So to talk more about the fixation chemicals, there are two main families of fixation chemicals out there for use in tissues. One is coagulating chemicals. We don't use this very much in tissue imaging experiments, because it doesn't preserve tissue morphology particularly well, and it generally has pretty poor penetration. This is often more used in flow cytometry experiments, but I'd like to mention it here as there are certain situations where it might be necessary. Often, however, what folks use for tissue imaging experiments is chemical cross-linkers. So the most common one is formalin, or neutral buffered formalin, which is 10% formalin in PBS. This is actually a mixture of formaldehyde and a little bit of methanol, as the methanol helps the formaldehyde not oxidize and form a formic acid, which isn't very good for your fixation. If you would like to avoid using methanol for whatever reason, you can actually use straight or use a 4% paraformaldehyde as long as it's mixed fresh just before use. As the MBF ages, it can become acidic and it can cause some background effects. Another fixation chemical is glutaraldehyde. It causes stronger protein crosslinks, but it can extract soluble antigens and modify tissue architecture. It can also cause increased autofluorescence. So some of the considerations you want to take into account when choosing your fixation type is what kind of antigens you're trying to detect and whether or not you're going to be using antibodies. Certain antibodies have suggested fixation types that work best for them, and it's worth looking at your product's literature before you make a decision. Another thing to consider is if you're using a fluorescent detection modality, think about the autofluorescence. So you might not want to use glutaraldehyde if you're thinking of using a fluorescence detection modality. I'm going to say this a lot in this presentation, and I apologize in advance, but you're going to have to optimize things in your own hands as well. So fixation times, you can underfix and you can also overfix. In any case, that might result in non-optimal staining. So there are situations where you can go to the literature and find what best practices are, but you still might have to run some experiments in your own hands to optimize. When dealing with tissue, it's always good to examine the tissue prior to use, making sure that it's not bad tissue quality, it hasn't been damaged or mishandled or misstored. Sometimes tissue is poorly adhered to the slides and you'll start noticing it coming up off the slides. It is important to identify this early in your experiment so that you don't use expensive reagents and waste time on tissue that is going to give you poor results no matter what you do. Once you have rehydrated your tissue, don't let the tissue dry out. This can kind of rehide your epitopes and antigens and may cause inconsistent staining. So once the tissue has been rehydrated, make sure it stays moist. When working with tissue, of course, it's important to take precautions against damaging your tissue simply by handling it. You really want to keep track of which side of the slide your tissue is on, and so that you don't accidentally wipe it off or mush it with a finger. You want to make sure that when you're working with forceps or pipette tips that they aren't coming in contact with your tissue causing scoring damages. Often it helps to mark the tissue side of your slide or make notes about your experimental conditions on the writing portion of your slide, that white part you can see up at the top there. I often use pencil instead of any kind of marker because even solvent-resistant markers can sometimes start washing away if you're using strong solvents. If you have reagents that are expensive or precious, you can also minimize volume while avoiding tissue drying out by using cover slips. So you can use a smaller volume and then gently lay a cover slip across the top of your reagents and your tissue to help minimize that headroom and really allow you to use smaller volumes. When doing this, it's important to angle the cover slip and lower it slowly so you don't have air bubbles, which can also cause inconsistent staining patterns. It is important to practice this. You'll find that it's a lot easier when you do it multiple times. When working with FFPE tissue, one needs to remove the paraffin that the tissue is embedded with and rehydrate it in order to work with most detection modalities. This involves soaking these slides in a rehydration station, essentially. So you start with xylene, which helps remove the paraffin, and then you use a graded rehydration series of ethanol and water to first remove the xylene, and then rehydrate this tissue to get it back into solutions that are can be used for antibodies and other detection strategies. So I guess polar. I strongly suggest using fresh solutions every time you do this, for knowing the percentages of the various buffers is important and can really make a difference in terms of removing all of the paraffin and making sure your tissue is well rehydrated. When you are doing this, it's important to treat your slides gently. So you will want to agitate the slides in these solutions, but you want to do it gently because you don't want your tissue to come loose in these solutions either. Agitating is important to remove all of the paraffin and rehydrate your tissue well, but again, gentle. So many common fixation strategies involve chemical cross-linking, where your protein is chemically cross-linked across to help basically stabilize it long term. However, when that happens, it can hide epitopes or antigens of interest. So often there is an epitope or antigen retrieval step. This is very common when working with FFPE tissue. It can be a little bit less common with cryosections, depending on what antigen or detection strategy you're using. There are two common ways of epitope retrieval. One is heat-induced epitope retrieval, and the other is protease-induced epitope retrieval. Heat-induced epitope retrieval involves using a common buffer, often citrate or EDTA. This is another one where you're going to have to do some research about the antigens you're looking for or what detection strategies or antibodies you're using to see what they suggest. You place your slides with the in these buffers, and then you heat them to a high temperature. These can involve water baths or even stir plates. Another common method is to actually use a pressure cooker. This allows the buffers that you're using, the antigen retrieval buffers you're using, to get to higher temperatures, but makes it so that the buffers won't boil. You do not want to boil your tissue. This can cause inconsistent staining because you are essentially causing weird heating patterns atop your tissue. This can also remove your tissue from your slides because you literally boil it into your solution. So do not let your solutions boil when doing heat-induced epitope retrieval. Again, you're going to have to optimize in your own hands. Overcooking can be just as bad as undercooking when it comes to antigen retrieval. I have an example here where in one situation, the tissue was placed in a pressure cooker and cooked for 10 minutes. And then to the left, we have one where it was cooked for 20. And you can see that by 20, we're actually seeing the staining, we're getting good response, but at 10, the antigens just haven't been fully retrieved yet. Another method of epitope retrieval is protease-induced epitope retrieval. This is very common in situ hybridization experiments. And so what you use is actually proteases like protease K, trypsin, or pepsin to do a similar thing as the heat-induced epitope retrieval, but by protease. So it basically eats away a little bit of the protein to help relax these chemical crosslinks. It is, of course, you're going to have to optimize for your experiments based on your antigens and what kind of detection strategy you're using. It is absolutely possible to over-digest just as easily it is to under-digest. So I strongly suggest running a couple experiments to find your optimal conditions. When using any detection strategy, it's important to block and neutralize off-target detections. So each detection modality has kind of its own complications. We'll be going into some of these in more depth, but there are many things to consider. For example, there are endogenous enzymes and proteins in tissues that can cause off-target detections. Other things to consider are, for example, if you're going to use alkaline phosphatase as a signal amplifier in your detection strategy, you don't want to use phosphate buffered saline solution in any of your buffers. The phosphatase and the, and the PBS means that you can't really get good signal. Another thing to consider is if you're detecting any kind of nucleotides like RNA or DNA, you might want to include some kind of RNase or DNase inhibitor so that the biomarker you're trying to detect isn't being degraded while you're trying to detect it. So when working with antibodies, it's important to use a blocking reagent so that the antibody doesn't stick to very sticky parts of the tissue and only adheres to the antigen of interest. Common blocking reagents are basically protein solutions, especially normal serum. There are also solutions of BSA, gelatin, and non-fat milk. Normal serum is the serum from the host species of the secondary antibody and carries antibodies that bind to reactive sites and allows for us to prevent nonspecific binding of the secondary antibodies. It is important to optimize these blocking steps by using positive and negative controls so you can monitor both the background as well as your signal strength. And then you can choose a blocking buffer that yields the highest signal to noise ratio. You can also use the same blocking buffer to dilute your antibodies solutions. Invitrogen or Thermo Fisher has a lot of really good blocking reagents that can be a very good place to start when you're starting out with this kind of science, including blockade and a couple others. You want to ensure that there's no substance in the blocking buffer that will interfere with this particular assay. For example, don't block with non-fat milk if you're using any kind of biotin streptavidin detection strategy, because non-fat milk contains biotin. So, as I mentioned earlier, there are endogenous signals in certain tissues that you'll want to account for if you're using specific signal amplification strategies. For example, if you're using antibodies that have or secondary antibodies or any detection strategy that involves horseradish peroxidase, you want to make sure that you've quenched the endogenous peroxidase in your tissues. If you don't, you might end up with off-target signal from these endogenous enzymes. This can also happen with alkaline phosphatase, where there is you can quench them with acid or lamidazole. There is also Ready Probe's endogenous HRP and AP blocking solution that I strongly suggest. It's a one and done solution where you don't have to think about this nearly as hard. Another thing to consider is, of course, that some tissues have endogenous biotin in them and need to be neutralized if you're using a streptavidin detection system. And again, Ready Probes has a solution for that as well. If you're doing any kind of detecting in a fluorescence channel, you really need to consider the autofluorescence. There are many sources of autofluorescence, including the fixation methods, if you've used glutaraldehyde, but even just normal FFPE with paraffin can cause increase in autofluorescence. There are also many natural sources, structural elements like collagen or any repeating element in the extracellular matrix. There's also plant proteins, pigments, red blood cells. All of these can cause autofluorescence that increase the amount of background fluorescence, making your true signal harder to detect. When neutralizing autofluorescence, there's a couple different strategies. One is literally quenching the autofluorescence. So Ready Probes has a tissue autofluorescent quenching kit that I, again, strongly recommend. You can see here on the right, there's the non-quenched as image A, where the collagen in this lung tissue is incredibly autofluorescent. After using this kit, it decreases to the point where you can barely see it. I've used this many times and I cannot recommend it enough. There are other strategies, of course. You can amplify over the autofluorescence. So you can use something like a tyramide amplification to increase your true signal over the background enough that you can distinguish one from the other. Another option is to digitally remove the autofluorescence. So using a software like Celeste or any kind of spectral deconvolution system, you can basically suppress the signal from the target, off-target background noise and increase the true signal. So when working with various detection strategies, one of the other things to consider is your imaging and your mountant. So based on your microscope and objective needs, the focal depth and the sharpness of the image that you're trying to get, it's important to consider all of these when choosing mountants. So if you're using some kind of fluorescent detection modality, you might want to consider whether you need an anti-fade reagent. Are you concerned about photo bleaching? That being said, if you're doing some kind of cyclical staining where you intend to photo bleach in between your staining cycles, you might not want to include an anti-fade, as that might make that more complicated. One of the things to consider when choosing mountants is the refractive index matching. If you match your refractive index to your microscope and objective needs, you can do sharper, deeper imaging with less effort. When choosing whether or not you're going to mount, there are many considerations. For example, if you're doing in situ hybridization that has these sharp, distinct puncta, often this kind of detection strategy really benefits from a mountant, so you can get these nice clear images. However, some antibody staining is broader, more localized, and might not need that clarity to get good data. If you're doing some kind of cyclical detection, sometimes you can do something called unmounted, which means basically imaging in buffers instead of using a true mountant. There are various imaging chambers as well as temporary cover slip options, where essentially you just lay the cover slip down on top of your tissue, include an imaging buffer, and then image through that. This allows for this to be easily removed so you can go on with your cyclical staining. However, if you need a mountain, there are two main sorts. One is organic. This is based on solvents like toluene and xylene. So this organic mountains aren't compatible with some detection modalities. It's not really suggested for most fluorescent detection modalities, and even some colorimetric detection modalities are incompatible with organic mountains because they're based on precipitation chemistry. And if you apply any solvent, they will wash away or get blurry. If you're using a fluorescent detection modality, aqueous mountains are really the way to go. Invitrogen has two different sorts. There's prolong and slow fade. Prolong is our hard set mountain, and so if after it dries, the cover slip just really will not move. It also contains anti-fade reagents so that you have to worry less about photo bleaching while you're imaging. And it has very good RI matching capabilities that will give you that depth and clarity in your imaging. So another thing to consider when you're planning out these experiments is controls and, of course, optimization. Let's start with controls. You will want both negative and positive controls whenever possible. When you're starting out, sometimes it's difficult to have a positive control, but you will always want negative controls. One good control to have to start is an autofluorescent control, which is a control where you have processed your tissue, but you have not applied any detection modalities. This will tell you whether you have a lot of autofluorescence to deal with and whether you should use some kind of quench. A no primary antibody control is also strongly recommended. This means if you're using any kind of antibody detection, you simply do not use your primary antibody in that sample. You still use all your secondary methods, and this will give you a chance to see if there's any non-specific or off-target signal. There are also things called cross-label controls. These are if you're using antibodies from multiple species in a multiplex experiment. These are a little bit less relevant these days, especially if you use good validated antibodies. But if you have any concerns, a no cross-label control can show you that your primary antibodies are only interacting with the secondaries that you anticipated them reacting with. When it comes to optimization, you've heard me say many times in this webinar, and I'll say it again, optimization is critical to get your best data. There are many different steps at which you can optimize. You can optimize your fixation in terms of how long you fix and what chemicals you're using. You can optimize your antigen retrieval, again, optimizing the amount of time, the heat, or even the buffer. Incubation times are also very important in terms of how long you're incubating with your various detection modalities like your antibodies or your probes. And of course, it's always important to consider the order of the detection modalities and whether or not they can be interacting. For example, if you're going to be doing ISH and antibodies, you might want to consider how the block step that you need for good antibody signal might affect your in situ hybridization detection. This is where using known working reagents and protocols can be incredibly useful. I strongly recommend using validated antibodies because often they will have good suggestions about what fixation methods to use, how long you should be incubating, and what concentrations to use. Look at your product sheets for the various detection reagents you're intending to use and read them carefully. It is also important to look at literature, databases, and others' protocols. Sometimes you can get ahead by using someone else's working protocols in order to base your own. So when it comes to multiplexing, there's a lot to keep in mind, but considering these out ahead of time can make for better data in the end. Again, order can matter. So consider how the various detection modalities might interact. One thing to also consider is that if you're considering using a colorimetric detection strategy and a fluorescent detection strategy in one sample, many colorimetric substrates have fluorescent profiles. So for example, eosin of our favorite H&E stain is highly fluorescent in the green channel. So you will not want to use eosin staining if you're considering detecting anything in the GFP channel. Another thing to consider if you're going for cyclical detection is how you might remove this signal. I'm not going to go over this in detail today. There are following webinars that will be coming up in this series that will touch on this more. But one thing to consider is that if you are doing cyclical detection, considering whether your more robust signals versus things that you consider rarer or weaker. Understanding where you should put them in terms of your detection strategy can result in much clearer and better data. So in this presentation, we've talked a lot about a lot of various considerations. So right now, I'm going to run us through a quick thought experiment just to kind of illustrate what I've been talking about. Oops, pushed audience. So in this example, we have FFPE retinal tissue that we're going to run detections with antibodies, a primary antibody and a series of secondary antibody conjugates that we're going to use fluorescent. We're also going to stain with a nuclear counterstain and do a single round of detection. So what are some of the things we should consider? Well, we've already said that we're using FFPE tissue, so we'll be wanting to deparaffinize and use some kind of epitope retrieval. In this case, I think I'm going to use heat. We've talked that we're going to use antibodies and a nuclear counterstain, so we'll consider those as well. Since we're using antibodies, we'll want to use a protein block so that the antibody does not have off-target staining. And we said that we're going to use fluorescence, so we'll want to consider an autofluorescence quenching step. We won't be using any signal amplification, and we're only using one round of detection, so I'll be using the aqueous hard mount. In terms of our control, we'll use a no primary antibody detection, and I might consider an experimental order control if I'm concerned about my nuclear counterstain. So here's all our considerations highlighted. So let's look at our experiment. So we had our FFPE tissue. We paraffinized, antigen retrieved, permeabilized so that the reagent could penetrate better. We blocked so that our primary and secondary antibodies wouldn't have off-target effects. And then we neutralized some autofluorescence. We then added a common nuclear counterstain, in this point DAPI, and then mounted using prolonged glass that has an anti-fade reagent. And then we imaged. And you can see here we have this beautiful multiplex image showing the beta tubulin and various neuronal cells that were stained with NuN, a neuronal biomarker. So, in conclusion, spatial biology provides combined molecular information and spatial resolution. This allows us a deeper understanding of normal tissue function as well as disease pathology. Tying these various detection modalities together can deepen and enrich data and research. Of course, it's complicated. There are a lot of considerations to take into account, multiple aspects to consider in multiplexing, and especially when using the multiple detection modalities. Good tissue handling and preparation served as the basis for good detection and good data. So considering these things out ahead can save you a lot of heartache. So in conclusion, don't be scared of multiplexing. It can be incredibly useful. Thank you so much for your time, and I'll be taking some questions.
All right. Well, thank you so much, Nancie, for your informative presentation. We will now go ahead and get started with the live Q&A portion of the webinar. So if you have a question that you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. All right, so let's get started. Nancie, we've already got some great questions coming in from our audience. So we'll start with this one here. Can you recommend a human tissue bank?
Sure. So for my work, I used a lot of tissue from a biobank called Ziogen. I've also used tissue from Amis Bio and BioChain. Often you can look at the other thing to consider is there are often, if you've got publications that you're interested in or data that you're trying to follow up on, sometimes they will tell you what supplier they were using. And that's kind of a good way to see that the tissue worked in their hands and kind of give you some confidence.
Great, thank you. Another question here asks, what will have less fluorescence background, cryopreserved or FFPE?
Often cryopreserved has much less background because it's a lot easier to control the fixation method. And you can find that it's a lot easier to tailor it. It's also kind of been shown that the longer it's been fixed, the longer your FFP has been sitting there in the paraffin, the autofluorescence just kind of gets set in. So in general, cryopreserved tissue has much less autofluorescence than FFP.
Great, thank you. This next question asks, do you have any suggestions for using non-hazardous deparaffinization reagents? Our lab is looking to remove xylene from our workflow.
Sure. So that's a common ask right now. A lot of people are trying to move away from these really hazardous solvents like xylene and toluene. One really common one is a histoclear. There are other solutions like that in the market. You can basically search for xylene substitutes. They involve a slightly different protocol, but often can, with again, a little optimization, can result in equal amounts of deparaffination as a xylene workflow.
Great, thank you. All right, and like I said, we're getting some great questions in from our audience, so we have time to continue with a few more here. This next question asks, with HIER, is there a trade-off between epitope retrieval and background labeling? In other words, does HIER also amplify non-specific autofluorescence labeling?
Sure. For the most part, HIAR won't increase autofluorescence per se. And as long as you've optimized it, it shouldn't increase your background labeling terribly much. If you've kind of underdeveloped or under antigen retrieved, you can end up with situations where your antibodies basically getting caught in like non-opened up protein goo. And then there's the other side where if you overcook, you can render your tissue so ravaged that anything will stick. So again, I have to lean on the whole optimize in your own hands. But if you do a good optimization, your HIER shouldn't increase your background terribly much.
Great. Now, this next question is a two-part question, and it asks, any suggestion for good protocol for immunohistofluorescence? And the second part asks, which antibody and which fluorescence would you use together?
So I'm going to want to reach out to this person offline to get a couple more questions answered in terms of what kind of experimental setup, what tissue they're using. But in general, on Thermo Fisher, we have some really good base protocols for a lot of common immunohistochemistry and immunofluorescent labeling that it can be a really good place to start. In terms of which antibodies and which fluorescent to use together, that depends a lot on what experiment and what targets, what tissue you're using. But in general, I have to suggest Thermo Fisher verified antibodies. As long as you know what your target is, it's really valuable to know that they've been appropriately screened. And so you have a lot less concerns about off-target effects or whether or not it's actually detecting the epitope or antigen that you're actually looking for. But I'll be reaching out so that I can get some more information about what exactly you're looking for.
Great, thank you. Another question I have asks, can I prepare 4% formaldehyde fixative solution in PBS from 16% paraformaldehyde solution in PBS?
Sure. So instead of using a neutral buffered formalin solution, you can absolutely use a paraformaldehyde solution. A lot of people do make up 4% solutions in PBS from a 16% solution. Just mind your PBS concentrations, make sure it stays around 1x. And again, when it comes to paraformaldehyde, making it up fresh just before you're using it will get you better results than anything that's been left in the fridge for months.
Great. Another question here asks, can you describe or elaborate on the pros and cons between HIER and PIER and when to use PIER?
Sure. So I would say that HIER, so heat-induced epitope retrieval, is a little more common, especially if you're using antibodies. The concern with using the protease-induced epitope retrieval, if you're looking for proteins to be detected for your antibody, is the fear that you're basically dissolving away the protein that you're trying to look for while you're using it. So for the most part, I would say that heat-induced epitope retrieval is better for antibodies as a general rule. Of course, now that I've said that, there's going to be something that's very specific somewhere in the literature that's going to say that, there's this one edge case where one's better than the other, but, optimization in your own hands. When it comes to the protease-induced epitope retrieval, that's often used in situ hybridization. So when you're trying to detect mRNAs or specific DNAs, essentially, because at that point, you're often less concerned with your proteins. And so it kind of gives you an opportunity to kind of dissolve the proteins away from your targets of interest.
Wonderful. Thank you so much. All right. Next question here asks, would you recommend using the conjunction kits for IF?
Sure. So using the conjugation kits for immunofluorescence, it's got the good parts and it's another step you can always optimize. So one of the nice parts about using the conjugations is that you can basically, instead of having a two-step antibody detection system where you have the one antibody that's detecting your antigen of interest, and then a secondary antibody that's detecting the antibody that you just put down, you can basically conjugate those two systems together and have the antibody that's detecting your antigen have the basically the signal on it already. So it can be very valuable in certain situations. However, one of the nice parts of the two-step system, so the primary and secondary, is you actually can get a little bit of basically signal amplification out of it, because what you can have happen is basically multiple secondaries can find a single primary antibody. So there's benefits, there's drawbacks, and I'm going to have to go back to my good old standall of optimizing in your own hands, kind of understanding what you're trying to get out of your system. Thermo Fisher has a lot of fantastic conjugation reagents, and I'm pretty confident that we've got some really good pages to help kind of walk you through the positives and negatives. But I would have to reach out with more information, probably get some idea about what you're thinking about.
Great. Thank you. All right. We've got another question here, and this one asks, when would you use pressure cooker for epitope retrieval rather than HIER?
Sure. So the pressure cooker is a type of HIER. I really like using the pressure cooker because it's got some some benefits in terms of being able to retrieve at higher temperatures without having to worry about boiling. It's back to that whole PV equals nRT to bring us all back to basically high school physics. As you increase the pressure, you can increase the temperature. So I like it for the ability that I can use slightly less time to get the same kind of heat-induced epitope retrieval. It also means that I have to worry a little bit less about the boiling. That being said, you'll find a lot of people using a lot of very interesting strategies. I know people who use special kinds of, basically they're rice cookers or steamers that you can kind of get at like Home Depot or Best Buy, as well as honestly just water bath systems. So when would I use the pressure cooker versus not using the pressure cooker? Again, optimizing in your own hands. You can find that the pressure cooker will work better in certain situations and less good in other situations. And unfortunately, it all really depends on your tissue and what antigens you're looking for.
Great. Well, thank you so much, Nancie, again for your time today and your important research. That will conclude our Q&A session for today. But Nancie, do you have any final comments for our audience before we go?
Sure. Again, I'm going to wrap up with optimization is so important. It's one of those things where once you start doing this kind of science, you realize it's the steps that you don't even consider when you started that can really make all the difference, especially your incubation times and how you're retrieving your antigens. And of course, enjoy your science. This is supposed to be a certain amount of fun. So don't think that optimization has to be boring. You'll be surprised how gratifying it is where you find the perfect conditions that give you the best data.
Great. Well, thank you again, Nancie, for your time and your research. We would also like to thank Labroots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you had provided at the time of registration. This webcast can be viewed on demand, and Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. So until next time, take care, everyone. Goodbye.